In this lecture, we will discuss the properties of receptors as drug targets. Specifically, we will review quickly the function of receptors, talk about signal transduction, give some examples of classes of receptors, and then talk about the design of antagonists, which is the main application for the use of receptors as targets of therapy. So let's look in general how receptors work. These are proteins that receive chemical signals, typically from outside the cell. Most of them are located in the cell membrane. So what happens is that binding to a chemical messenger of some type results in a change of conformation of the receptor, and consequently, a message is transmitted intracellularly. And then, when the messenger dissociated from the receptor goes back to the resting state and the message stops. So one important thing to know about receptors is that in general, this step in which the messenger results in a message being transmitted intracellularly amplifies the signal. In other words, binding of one molecule typically results in a downstream cascade intracellularly that is multi-molecular. In other words, for example, binding of a single molecule of growth factor will activate an enzyme kinase intracellularly, which then amplifies the signal by phosphorylating many more proteins. So we'll talk about how receptors are localized in the cell, and as I said, most of them will be on the cell surface. Some of them will be intracellular as well. And in general, each cell has a range of receptors in the cell membrane, which makes it responsive to different kinds of messengers. So what do we mean when we talk about a chemical messenger? A chemical messenger can be of various types. The most frequent example will be a neurotransmitter, which is a chemical released at the end of a neuron in a synaptic space, and then in turns this activates the neuron downstream from the first one, or the muscle cell downstream from this first neuron. And so typically the signal due to this kind of messages is short-lived and basically mediates cell-to-cell communication. Then we have hormones which are released in the bloodstream and basically reach another effector cell far away from where it has been secreted. So typically, we'll have a receptor that is activated by, for example, adrenaline, which is released by the suprarenal cortex in, above the kidney. So another important thing to notice is that messengers are not altered chemically by binding to the receptor. And we can divide receptors in several superfamilies: ion channels, G-protein coupled receptors, kinase link receptors, and intracellular receptors. The first three types are membrane bound, so they're found on the membrane of cells. And what's different between these enzymes is the response time. In other words, ion channels are activated immediately with a microseconds time scale. G-protein coupled receptors are activated, so basically they generate a downstream signal within seconds. And finally, kinase linked receptors and intracellular receptors, which typically act on protein synthesis, can have time of activation as long as minutes. So let's have a short survey of how these different receptors work. For example, ion channels are transmembrane proteins that control the influx of ions. Each type of ion channel is specific for one specific ion. So, for example, we have sodium, calcium, chlorine, potassium channels. And the ions flow across the cell membrane, you know, using basically concentration gradient. So, you know, basically they follow the concentration gradient. They go from the most concentrated compartment to the least concentrated compartment. And this is a typical mechanism that we have to polarize or depolarize nerve membranes. And in terms, you know, the change of the concentration of a certain ion within the cell is linked to the activation or other enzymes or other actions that are then transmitted intracellularly. So what happens is that normally in the resting state, the ion channel is closed, and then upon binding to a messenger, which is located typically close to the opening of the channel, we have a rearrangement of the channel, opening of the ion channel, and then basically passage of the ions. One example of that is the nicotinic receptor. The nicotinic receptor is formed by 5 transmembrane subunits, 2 of which bind to the neurotransmitter, which is basically acetylcholine, or also nicotine, which is obviously not endogenous. And what happens then, you know, is that we have a rearrangement of the channel. So in general, ion channels are linked to are used basically for neurotransmitters such as acetylcholine and GABA. And so they have a very fast response. So let's look a little bit more in detail at the structure, again with a cartoon. Each subunit of the nicotinic receptor is made up by 4 different transmembrane helices. Both the N and the C terminals are located on the extracellular portion of the cell membrane, and the neurotransmitter binding site is here close to the N terminus. The structure then looked for above is, you know, we have these 5 subunits related. Each of them is formed by these 4 transmembrane helices. The so-called TM2, so the transmembrane helix 2, ends up lining the pore of the channel, and so it's the one in charge of controlling passage of ions. And ion channels typically exhibit a behavior called gating, in which we have an opening of the channel upon binding of this messenger. In general, ion channels specific for cations, so for positively charged ions, are excitatory, and so they respond to neurotransmitters such as acetylcholine or nicotine, whereas anionic channels for chlorine typically respond to rather neurotransmitters such as GABA, which is inhibitors. So again, we have this upon binding of the messenger, we have gating, so opening of the channel, and free flux of the ions across the concentration gradient, so from more concentrated to less concentrated. This is a more in detailed cartoon of what happens exactly and how these transmembrane helices change their conformation slightly by rotation to allow for passage of the ions. The next class of cellular receptor is actually the most important one in therapy, and those are the G protein coupled receptors. This mediate most of the physiological responses to hormones, to neurotransmitters, to environmental stimuli, such as cold, for example, or light. They involved proteins that contain 7 transmembrane helices with extracellular loops that are in charge of recognizing the stimuli, so the neurotransmitter or the hormones that act on the channel. And then in the intracellular portion, they have a binding site that basically accommodates the G protein. So one important thing is that, for example, these receptors also respond to light. So rhodopsin, which is a membrane protein responsible for our ability to see, respond to is also a G protein coupled receptor. So in general, these G protein coupled receptors are the target for about 30% of the drugs currently on the market. And one of the complications was that until recently, it was almost impossible to know what they looked like. However, in the last 10 years, we have had advances in structural biology, and so we have been able to solve the structure of some of the membrane channels. The G protein coupled receptors, such as the beta adrenergic G protein coupled receptor. Another one, like rhodopsin, has been known for a long time, but the other G protein coupled receptors were elusive. And actually, one of our colleagues here at the ECU works extensively on the structure determination, in this case by NMR, of G protein coupled receptors that respond to temperature. So in particular, the channel he works on responds to cold and also to methanol. So interestingly enough, the sensation of freshness that we have when exposed to menthol is due to the fact that the same receptor that responds to changes in temperature to, basically to the cold, is activated by menthol. So what happens in this case? In the case of G protein coupled receptors, we have that the messenger induces a change in conformation intracellularly that then accommodates G protein. In turn, G protein then can give out downstream signaling, for example, by breaking into subunits and then in turn activating enzymes downstream. So the signaling is actually quite complex. And in general, these GPCR receptors can be either depending on G protein or not, with the same conformation. And so what happens in this case is that there's an intrinsic activity of the channel that responds to other proteins. So this slide is a little complicated. Let's walk through this in the next video.